I am super excited to join the section and the center, and even more so when I can meet you all in person. But I will talk a bit about the work that we are doing now and what I have been doing in the past. So microbes, as you know, are everywhere, and when they're in and on us, they can affect our health. So a lot of the work on the human microbiome has focused on identifying good and bad microbes, but we know that it's rarely that simple. So the effect of our microbes depend on the environment and context, and part of that context and environment are interactions with other organisms, social interactions. Much of what we know about social evolution has its foundation in social insects research, and that's also what I did for my master's and PhD. So when we think of ants and bees, we can easily envision different types of interaction, such as cooperation within an ant colony, and for example, conflict within a species in a bee colony over who gets to become the new queen. And they can, of course, also have a competition between different species. So we can see how these are important to both the species and the larger ecosystem that they're part of. But microbes also have more complex social lives than they're usually given credit for. They can interact within a population, between different species, and of course, with the host. As a PhD and master student at the Center for Social Evolution here at the university, I worked with interactions between microbes and ants. So I looked at Wolbachia, that are intracellular bacteria within the ants, but potentially actually also extracellular as one of the first insects that has been found in. I worked with Pseudomonas bacteria that grow on leafcutter ants. And the ants use these bacteria to produce antibiotics that they use as a pesticide in their fungus gardens. And we could show that diversity of these bacteria on individual ants and colonies is very low. So they basically only have one strain of bacteria. And that's probably a way to keep a good relationship between the host and the microbes, lowering competition between the bacteria. I also worked with this beautiful system with the zombie ants, where we can see that a fungus can infect ants, take over their brain, and manipulate their behavior so that the ants die in a spot that's good for the fungus. And this looks quite dramatic, but what we actually could show was that the social life of the host, that they're living in well-protected colonies, actually means that the cost at the colony level, the superorganism level, is very low. But as wonderful as all of these ants and tropical fieldwork is, I wanted to apply my evolutionary training to problems of more clinical relevance. So evolutionary medicine is quite a young field, and here we use evolutionary principles to understand why we get sick and how to improve prevention and treatment of disease. So it's an interdisciplinary field that has already contributed to our understanding, for example, of the development of cancer and mental health challenges. And there's obvious potential to use evolutionary principle to understand our relationship with the microbes that affect our health. And here we can also actually use many of these principles that we know from how social insects interact with each other. So just like the ants can cooperate, so can microbes. So bacteria frequently cooperate by secreting compounds that not only benefit themselves, but also neighboring cells around them. So if we have a population of cooperators, they will do better at a population level than a population of non-cooperators. However, when we mix them together, these non-cooperators may be able to exploit the cooperation of the others and actually increase in frequency to the detriment of the population as a whole and to the cooperators. So we understand these dynamics quite well from lab populations of microbes, but much less about what goes on in nature and especially in clinical populations of bacteria, clinically relevant bacteria. And this is quite important that we get a better understanding of this, because many of these exoproducts that bacteria can produce can affect our health. either as toxins, as virulent product, or as compounds that, for example, we can use in nutrition. So as a postdoc, I worked a lot on Pseudomonas aeruginosa, which is a gram-negative bacteria that's found ubiquitously in soil and water, and it has a large metabolic range. It's a common opportunistic pathogen, and it's also the star of many social evolution experiments. And a lot of this work has been done by Ashley Griffin and Melanie Gould at the University of Oxford, where I started my postdoc. So we understand social interactions of Pseudomonas quite well in the lab, but not what goes on in natural and clinical populations. So Pseudomonas is a big problem in cystic fibrosis patients. So cystic fibrosis is a genetic disease that causes the buildup of a thick mucus in the lungs that then get colonized by bacteria from the environment. And patients are colonized early on, treated with a lot of antibiotics, but the bacteria can be recovered later on, even though initially it was thought that these treatments would eradicate bacteria. But the bacteria regrow, and then we can see over time that they acquire mutations and they change. And one of the things that we often see is that a lot of these social traits of Pseudomonas are lost in chronic infections. So what drives this within host selection? It has primarily been thought that all of these changes are adaptations to the host environment. So changes in resource availability and of course antibiotic treatment. And this would imply that evolved lines have higher fitness in the host than the ancestor. However, we wanted to ask if social adaptation to intraspecific interactions also played a role. And here we might actually see that evolved lines have lower fitness in the host than the ancestor. Here I had access to a fantastic collection of cystic fibrosis isolates from many patients that are colonized with different clone types from the environment. And these patients have been sampled longitudinally, so we can see what mutations are rising, whether these mutations are retained in the population, and we can compare different clone types across patients and see if we find patterns of convergent evolution. So this is a collection that has been maintained by Helle Krogh Johansen and Søren Molin at DTU and Rigshospitalet, and Rasmus Lykke Marvin has done the genetic analysis of these bacteria. So what I focused on was the iron uptake systems in Pseudomonas aeruginosa. So almost 6% of all genes in Pseudomonas are their expression is dependent on iron limitation or a response to iron limitation. And what another interesting thing about these different systems for iron uptake is that they can be both cooperative or private. So I forgot to say that that iron is an essential nutrient that's often limited, especially within a host. So this is one of the ways that the human body try and limit infection is by withholding iron from pathogens. And it can do this by producing transferrin molecules, for example, that bind to the iron so that the bacteria can't take it up. So Pseudomonas has different ways around this. And one of them is by producing a siderophore called pyroverdin. So pyroverdin is secreted into the environment where it has a very high affinity for iron so that it can basically steal iron from host carriers. So it binds to iron and then it can be taken up by a specific receptor. And so this is a cooperative trait because once pyroverdin is produced and released into the environment and binds to iron, it can be taken up by any bacteria that has the right receptor. One of the other systems, however, is private. So this is the Pseudomonas heme uptake system, the FU system. And here we just simply have a receptor on the surface that can take up heme directly. So these two systems, we see quite a lot of changes over the course of an infection in the cystic fibrosis patients that I will summarize here. So we did phenotypic assays, and we looked at the mutations accumulating in the patients. So very briefly, what we saw that wild type isolates colonize the patients from the environment. And then we can either see one of two things happening. So either we see an increase in cooperation, so the bacteria start producing a lot more pyroverdin. And if this happens, then we are very likely to have an invasion of cheats. So that is bacteria that don't produce pyroverdin, but have a functional receptor that can steal the pyroverdin and the iron that the other bacteria are producing. If we don't have an upregulation of cooperation, we are less likely to see an invasion of cheats. We don't know why the bacteria upregulate cooperation in the beginning, but this could either be because iron is limited in the environment, or it could be also a way to compete with other bacteria, such as Staphylococcus aureus, because the staph don't have these specific receptors. So basically, if Pseudomonas bind all the iron in the environment to pyroverdin, then staph can compete on the iron. If we see an invasion of cheaters, we might see a complete loss of cooperation at the population level, so that we cannot sample any pyroverdin producers. So what happens next? Is this game over for the bacteria? No, we see an upregulation of the private system. So that this means that the bacteria are better at taking up heme directly from the environment. And this does not happen if cooperation is not lost. So with this analysis, we believe that we've shown that social interactions within a population of bacteria of the same species, interspecies interactions, are important even in a clinical environment. And this is something that we should take into consideration when we are developing strategies to treat these patients. This is quite a unique system where we have access to so many longitudinal samples. So some of these bacteria have been sampled over 40 years across patients as they've transmitted in the population of cystic fibrosis patients. So that's quite unique that we have access to this and the genomic information. But this system isn't ideal to look at how this affects the host, because there are no good mouse models for cystic fibrosis. And I really wanted to test the hypothesis that these intra and interspecific interactions between bacteria can also affect the host and the symbiont fitness. So what we're working on now is we're focusing on Helicobacter pylori and its complex relationship with the human host. So Helicobacter pylori lives in the human stomach, and it's an ancient association, as Helicobacter is thought to have co-evolved with modern humans. It's estimated that half of the world's population is infected, though with a big geographic discrepancy in where these people are infected. So in Europe, it would typically be less than 10% of a population that's infected with Helicobacter. When Helicobacter was discovered, it was found to be the causative agent of ulcers and cancers. So it was somewhat controversial when it was found that Helicobacter can also protect against other diseases such as asthma and allergies. But this is perhaps less surprising as an evolutionary biologist when you think of this, considering how long we have co-evolved with this organism. So what determines the outcome of an infection? So Helicobacter produce different compounds that interact with the host tissue, and production of some of these is positively correlated both with the risk of cancer, but also protection against asthma. So we can really think of these as host interactive compounds that drive the outcome. We know that the match between host and bacterial genetics also matter, and also the host age and infection. So most humans are infected early in life, and we know that here this can drive a tolerogenic response that also affects how the human will respond to other diseases later in life. So my mission is to try and use this system to look at both specific interactions within the Helicobacter population, look at how Helicobacter interact with other bacteria in the distal gut, and how it affects the response to other parasites. So how does Helicobacter interact in the stomach? So from looking at the genetics of the bacteria, we can see that there's a lot of horizontal gene transfer going on. You can have multiple strains of Helicobacter in the stomach, and there's also mutations accumulating over time. We hope to look at some of these interactions using organoids. So these organoids are advanced 3D cell cultures that present a major breakthrough in biomedical research, but hasn't really been used much in evolutionary studies. So from tissue from adult individuals, this could be mice or humans, you can isolate adult stem cells, and with the right cocktail of growth factors, these can develop into mini organs, in this case, stomachs, that have all the different cell types that a real stomach would have. So these can then be infected with Helicobacter. And this technique has primarily been used to look at what the effects on the host tissue are of infection. But we want to try and use this to look at interactions between wild-type Helicobacter and knockout of specific genes that are important in the host interactions. So hopefully by doing this, we can get an understanding of what's going on within the Helicobacter population, and then we can translate this into our mouse experiments. So C.A. Clew is a PhD student who will also join the section, and she's looking at how crosstalk between Helicobacter and the gut microbiome affect host metabolism. So we know that Helicobacter infection has both direct and indirect effects on host metabolism. In the stomach tissue, it can remodel the glands that are producing hormones that affect hunger and satiety. As I briefly mentioned, it can also affect the development of the immune system when you have an early life infection, and it can have systemic anti-inflammatory effects. It can also affect the composition of the distal gut microbiome, and this is likely also through the immune system. So what CA will do in her project is testing whether early life infection with Helicobacter can protect against diet-induced obesity later in life. So mice will be infected when they're 1 week old, and then they will be put on a high-fat diet, and then we will follow the microbiome composition, their weight, their accumulation of fat by MRI, and also look at the immune response. We can manipulate the composition of the distal gut microbiome by treating with antibiotics. And we can also introduce a mixed community of Helicobacter to see what the effects are of interactions within the Helicobacter community. So in this way, we can manipulate different parts of the system to get at the direct and indirect effects on host metabolism. So CA just started in May, but she's already been doing some 60-nest sequencing of the fecal gut microbiome. And what we see is that infection with Helicobacter, either the wild type or a knockout of one of these important host interactive compounds, lowers the diversity of the fecal microbiome. This is only seen at weaning. Already 1 week after weaning, the groups are undistinguishable. But we already know from other studies that such differences early in life may still have an effect on the host. So this is something that we'll be following up on. In collaboration with Torben Hansen at the Center for Basic Metabolic Research, we will hopefully also try and translate this by screening some human samples. So Torben has, for many years, studied metabolism and diabetes in individuals from Greenland. And in Greenland, we also have a high incidence of Helicobacter. So we will screen these samples to see whether we can correlate infection with Helicobacter with metabolic parameters. Katrine Graversen is a postdoc who will look at whether this effects on the immune system with Helicobacter early in life also affects how the host responds to a parasite challenge later in life. So historically and at present, there's quite a big overlap in the incidence of Helicobacter and malaria. So this could make it quite possible that our adaptation to one affects our response to the other. So malaria is caused by protozoa that's transmitted by mosquito. They travel to the liver where they multiply and then transition to life in the red blood cells. Rupture of the infected red blood cells cause disease, and we can also have both infected red blood cells but also white blood cells that adhere to the endothelia, and this enhances inflammation and cause obstructions. This is probably beneficial to the parasite as it avoids clearance in the spleen from the infected blood cells. And when this happens in the brain, we can see fatal cerebral malaria develop. So we want to use a mouse model on cerebral malaria to test whether the anti-inflammatory properties from Helicobacter can protect against cerebral malaria. So again, here we will infect mice with Helicobacter early in life and compared to uninfected mice. We will give them malaria later in life and then look at how well they survive, the level of anemia they develop, and the level of inflammation in tissues. So our pilot experiments already showed that if you have Helicobacter, infected with Helicobacter early in life, you're less likely to develop cerebral malaria later in life. And this is not because the levels of the parasites are lower, but likely because of the levels of inflammation. So here we can image the infections because the malaria parasites are bioluminescent. We can also count them by staining in red blood cells. If we look at the spleen right before the mice start developing cerebral malaria, by qPCR, I found that they have lower levels of inflammation. But Katrine has since done a transcriptomic that doesn't quite support this. So this is something we will follow up on. To translate this mouse work, we are also screening serum from malaria patients. And so far, I've only been able to get a hold of quite small collections of samples. And here we've been looking at two different malaria parasites, where in one population we find a trend in the direction that people with Helicobacter are less likely to develop severe malaria, whereas for the other parasite, the trend is in the opposite direction. So this is something we would like to follow up on with larger collections of samples. And it's also quite interesting because these two parasites have quite a different length of association with humans, as falciparum has been infecting humans for a lot shorter time. We're doing this work with a lot of collaborators, both here in Copenhagen and abroad. And I'm generously funded by Lundbeckfonden and the Independent Research Council Denmark. Thank you for listening.